Hi, my name is Maren, and this is the first time I have this kind of presentation, so I'm very excited to get this experience. And as Sridhar said, I'm doing a bachelor in biology at the University of Bergen, and this spring I'm enrolled in two courses at UNIS, one being an intern with SIOS, which gave me the possibility to be a part of organizing this webinar. And the other being a course called Research Project in Arctic Biology. And by participating in this project, it gave me the opportunity to contribute to the larger project of BIG. So I will start presenting what BIG is. And since my project is still ongoing and we haven't dived into any analysis yet and made the figures that we want to make, I will focus on what our goal with the study is and how we are collecting data. So BIG, Bjørndalen Integrated Gradients, it's a project that was initiated by the Arctic Biology Department at UNIS to make a cost-effective field laboratory, since field work in the Arctic is both logistically challenging and costly. So by establishing measurement stations close to Longyearbyen and combining resources, BIG included all habitats from the terrestrial sites to the littoral and pelagic systems in Bjørndalen. This increases collaboration both within and outside of the department, and it creates an authentic research experience for students like me, because we get to participate in different aspects of research in the study systems in Bjørndalen. And the overall knowledge of the ecosystem will be increased this way, and with all the data collected available long-term will be collected available long-term in a common database, and it would eventually also be possible to separate seasonal variations from climate change. And some of the activities going on in BIG is that they have this model system for autopolyploidy research that were established using Saxifraga opustifolia to understand the evolutionary consequences of autopolyploidy. And they also have time-lapse cameras that enables automated monitoring of plant phenology and plant pollinator interactions. They are also conducting monitoring of vegetation changes over time to understand how the vegetation responds to the permafrost that is under threat in Arctic regions. And in 2019, a seasonal reindeer project also became a part of BIG. And since then, weekly counting has been done from February, March until September, October. So I'm working on the spatial ecology of the Svalbard reindeer, and I'm working with a hypothesis that we expect that the spatial habitat use of reindeer in Bjørndalen is related to changes in snow cover and vegetation emergence. So the Svalbard reindeer are the largest herbivore here on Svalbard, and they play an important role in the ecosystem. So they have no natural enemies, so their population are mostly regulated by bottom-up control. And due to poor vegetation access in winter, they're adapted to really utilize the short growing season, and they can storage large amounts of fat, body fat, before the winter. The last 50 years, there have been conducted a lot of research on the Svalbard reindeer, and there is good knowledge about their adaptations to this high Arctic environment. But what we don't know too much about is how the reindeer use the landscape seasonally. And by using Bjørndalen as study site, we want to investigate if there are a correlation between weather data and spatial distribution. So to answer our research questions, we want to gather data from several factors that can influence the spatial habitat use of the reindeer in Bjørndalen. And as I mentioned before, weekly counts have been conducted since 2019, and I have been doing that this spring together with a fellow student. So once a week, we drive through the valley on snowmobiles looking for reindeer through binoculars. And when we spot them, we move towards them so we can determine age sex within the groups. And we divide them into calves, yearlings, adult males, and adult females. But if we're not able to see them properly, the age sex determination is dropped and we file them as unspecified. So we also mark their coordinates on a map. So we can get their position for every group of the reindeer that we see. And these weekly countings can give an overview over the seasonal fluctuations in density of reindeer in the valley. And we can see if there are any patterns on the total amount of reindeer over time, and if this can be linked to vegetation access. Now we can already see that there are more reindeers in the valley than there were earlier this spring, and we can also see more males present. So when we began in February, there was no males at all. And what we also noticed is that they are now foraging way up in the mountains. They're so high up that they can be so difficult to spot, and you really have to scan the entire mountainside to be able to see them. And they haven't been that high up in the mountains earlier, and maybe that's because of the snow conditions. So we are also gathering information about the snow depth that they are foraging in. And we made 3 categories for the snow depth, where 0 is if there is no snow, and 1 if there is snow, but we can still kind of see their eyes when they are foraging. And then we made 2 as to if the snow is so deep that we can't see their eyes. And we also have snow data from the weather station at the airport, so we can see the development of snow cover over a certain period of time. But since these data are outside of the Bjørndalen area, we also made 4 snow transects at 4 different coordinates throughout the valley, where we measure snow depth for every 50 meters in the direction of the red dots on this map. And we made these transects only a few weeks ago, so we don't have data for the entire period that there has been snow in the valley, because this these measurements they haven't been conducted in Bjørndalen before. So this is something new for this year, that we want to link this together with spatial ecology. But there are also time-lapse stations in Bjørndalen that makes it possible for us to follow the snowmelt and vegetation emergence. So maybe we can use these as well to get an overview over the last couple of years and not just this spring. And eventually we want to analyze our data on snow conditions, on temporal observations, and the vegetation types that are in Bjørndalen, and to find out if there are any correlation between these data and the spatial habitat use of Bjørndalen. So based on our results, we hope to maybe look at how the winter precipitation and snow conditions affect the spatial ecology on a bigger scale, linking it to climate change or natural common weather phenomena that are affected by the North Atlantic Oscillation. Yeah, so this was a little bit about what I have been doing as a student here at UNIS, participating in this research project. And thank you for listening.
Yes, thank you. And good morning, everyone. I think I know quite a few of the people who are listening in. Nice to meet the rest of you. And thanks, Maren, for talking about the big project. I'm going to come back to that very briefly at the end. But as you said, this is a project which is going to, in principle, continue for the foreseeable future. So it's going to be a project where we gather a reasonable data set concerning the ecology, terrestrial ecology of Svalbard. What I thought I'd do today is quickly run through the sort of state of knowledge of the invertebrates in Svalbard, and also a few areas where we're going to try and start better monitoring and trying to get a better idea about what species are here, and also looking at fluctuations, long-term trends, patterns in the populations and the population cycles. So I'm going to come back to big a little bit and talk a bit about something called COAT as well. I only had 10 minutes, so it's not long, but I will try and get finished in that time. My clock decided it doesn't want to work. Okay. So, without further ado, we'll start. Now, most people, when they find I work with invertebrates in Svalbard, certainly those living in Longyearbyen, are a little bit surprised to find that there's actually anything here to study. And in fact, there is quite a lot once you start to look into it. In 2014, a group of taxonomists and ecologists wrote a review article looking at the invertebrate biodiversity of not only Svalbard, but also Franz Josef and Novaya Zemlya. And the idea was to, first of all, come up with an inventory of what species are present, but also look to see if there's any common patterns between these three archipelagos, which sort of border the Barents Sea. And I won't say much more about this, except that we came up with a list of around 1,000 species of invertebrates that are known from Svalbard. At least their names in the literature. And these sort of animals include things like the colembla, which is this bright yellow large animal in front of the in the middle of the screen. We have large numbers of mites, these little animals here, Oribatid mites. These are present in maybe 20,000 animals per meter squared in the soil. More species of colembla. These can be found in up to 400,000 animals per meter squared. We also have a few beetles. This one's a weevil that's found on Salix. these are found in quite low numbers, and the reasons for that are a little bit unclear, why we don't find more beetle species in Svalbard, but traditionally we don't find many beetles at high latitudes. And then we're going to hear more about the rather interesting species of aphid, and here we have one aphid feeding on a dryas flower. And at the moment we know of 3 species from Svalbard. One is actually unidentified, and the other 2 we believe are endemics. So I'm looking forward to hearing the next presentation concerning these. And then we have 3 species of moth, which may surprise some people because they're only found in quite low numbers, but these are present year-round in Svalbard. And then of course we have the flower flies, such as this one, which are involved in pollination and important for plant breeding success. But everything I've said comes from only 2 locations really in Svalbard, and I think Peter Convey is going to talk a bit more about this. I'm not going to hang around too much on this slide, except to say that we have around 600 different species or papers describing the invertebrates of Svalbard. Pretty much all come from Isfjord or Ny-Ålesund area. There's a few from Hornsund in the south. And I know of 3 papers from Edgøya, 1 from Barentsøya, and 2 from Nord-Ausland. But mainly what we know is from the eastern regions. And this perhaps isn't surprising because this is where the settlements are, this is where we're working. Working in the east is actually quite tricky. It's expensive, it's very weather dependent, and it's also larger areas of protected ground. So it's hard to get permission sometimes to work in areas like Edgøya and Nord-Ausland. So that's very important. So the question then is, do we actually think that the populations, the species, the communities on these other areas are going to be different from what we know from around here? And I just want to give a few examples to show that yes, they are. They are different, and they're also very interesting, I think. This is one nice example. This is a moth called Plutella polaris. It's a relation to Plutella xylostella, which is a common vagrant found in Svalbard. Not every summer, but it's observed in most summers, maybe. It's a wind vagrant which comes to Svalbard if there are suitable southerly winds. However, this species, Plutella polaris, is resident in Svalbard. However, it was first observed in 1873, and 7 individuals were collected and stored in the Natural History Museum in London. And it was never seen again. These are the only individuals of this species. So there was a bit of discussion in the literature. Is this a genuine species? Is it an abnormal Plutella xylostella? And then partly because of the UNIS bachelor course, the Terrestrial Arctic Biology course, Geir got the opportunity to visit Ringhorndalen in Vedefjord, which is up here, where we believe the animal had been collected before. And he was able to trap this species. And yes, it is. It is definitely a species. So it hadn't been seen for over 130 years or so, 140 years. And now it's back in the list as a genuine species. At the time when he refound it, it was believed to be only known from Svalbard. Since then, it has actually been observed in Siberia. So it seems to be a high Arctic specialist that has very local populations. Moving quickly on, we can also say that if we go to Edgøya, we find new species. We were able to sample there thanks to the Arctic Field Grant. And although we could only go there for a very short period, flew the helicopter, landed, grabbed samples from around the soil samples from around the helicopter, came back and looked at what we got in those soil. We managed to find 140 different species, at least name to name, of which 16 hadn't been seen before from the eastern coast, from the western coast of Svalbard. So 11% were new records. So we have good evidence that the communities on the eastern coast are actually very different from the western coast. And that then begins to lead us to reappraise what we know about the invertebrates from Svalbard. And then the last example is this one, which is very unusual. This species had never been recorded from Svalbard until some samples, again taken by the bachelor course, were analysed by two mite taxonomists in Poland, Senexak. And in those samples, they found this species. Now, this species has only been seen once before. In Svalbard, we found it here in Mosselbukta, on the very far north of Svalbard. Just a few individuals, but enough for Anna to be able to identify them as Scutezetes clavidosensilis. Now, the interesting thing is that this species is only known from one other place, and it's here in Nepal. So we now have a species that has a distribution apparently in Nepal and Svalbard. Now, obviously, this isn't true. This is just indicates how little we know about the distribution of some of these Arctic, alpine, and Himalayan species. These species are perhaps adapted to low temperatures and high latitude environments. However, there is a campaign or a approach by the Conservation of Arctic Flora and Fauna to try and improve this. The next State of the Arctic Terrestrial Biodiversity report will come out in the next month or so. So I just wanted to quickly really inform people that this is coming. And just show that some of the data which are going to be presented in this shows significant changes due to what we believe is climate change. And the most comprehensive data we have for the invertebrates comes from perhaps Zackenberg in Greenland. And in this figure, I'm showing numbers of animals caught. This is the abundance against year. Oops, sorry. The years have disappeared behind the type text, but it's going between 1996 up to 2014. And you can see quite large annual variations, and this is characteristic of invertebrate faunas. The solid line shows a statistical decrease in the number of individuals of this one species. And in this line, we're showing a non-significant, but still a decrease, although you can suggest that this is partly due to the starting point. So we have to be a bit careful how we interpret some of these data. However, the point is that we're getting changes, possibly due to climate change. And these species, I should say, are flies, which are very important in pollination processes. But although we're asked continually to explain what's happening in climate change and environmental change in the Arctic, it's very difficult in the Arctic. We have very little monitoring. This is one of the conclusions of the report. Red shows where there is no monitoring of this, what's called focal ecological component. So these are species which are classified as blood feeding, pollination, prey for vertebrates, decomposers, herbivores, or predators. And red is no monitoring whatsoever. Green is there is some monitoring, but it's spatially very restricted. And the mode in this case is there is some monitoring, but its funding is ad hoc. So for the Arctic, there is pretty much no good financially secured monitoring of invertebrates. This is something we need to address. One way to address this is the big project, which you've heard nicely presented by Maren. This is going to be a approach by the Boris Department to work together to try and collect data which is of long-term use, time series. And as part of this, not only reindeer, we're looking at the invertebrates and we're trapping the invertebrates. There's also a project called COAT, the Climate Observatory for Arctic Tundra. And this has now started operating in Svalbard, with Åshild Pedersen as the leader. However, we have Roosbacker in the audience, and he is working full-time on this to improve the data collection, both for reindeer, but also looking at the invertebrates. So we're in the process of setting up invertebrate traps, which will, in principle, continue over an extended period and gather these long time series data we're lacking. And lastly, I would like to say that one of the real handicaps in invertebrate research is the time it takes to identify and sort samples. And so one approach that Jesper and I are both considering at the moment is getting these animals sequenced. And this table shows different groups of invertebrates. It shows how many species we believe there to be in Svalbard and how many have already been sequenced from somewhere in the world. So there's quite a large number of the Svalbard fauna, for example, Colembla, 71 species, 52 have sequences in bold, 73%. So what we need to do is complete the missing species in Svalbard, get them sequenced, and then we could perhaps think about enhancing the rate of identifications. We can think about perhaps sampling the preservation fluid, sequencing that and getting an idea of which species are present in the trap catch. And this will increase the speed that we can actually assess both the species which are present, but also the densities. And that's really very important with monitoring, because in monitoring, you have to not only collect the data, but you have to assess it and publish the data continually. You can't wait for 10 years before you start having time to analyze those data. And say this is a major stumbling block for invertebrate research. So with that, I think I'm running out of time, so I'll wrap up. And just really say that there's a very diverse fauna in Svalbard. It's a crucial component of the ecosystem, and it has a wide range of roles, including detritivores, pollination services, parasites, and prey items. But we know very little about the actual communities beyond those from Isfjord and Kongsfjord. And we have pretty much nothing on the trends and the patterns. And with that, I will thank you and hand over to the next two presenters.
Oh, hello, everyone. Thank you very much for this invitation. And so today I'd like to tell you a few words about aphids as an interesting group of insects, and in particular about the amazing aphids of Svalbard. The known world fauna of aphids recently reached more than 5,000 species, and is recorded from about each in species. In comparison with other hemipterans, as you can see, is very diverse and known by interesting and unusual phenomena. Among others, the reproductive strategy of aphids seems to be a clue in understanding their evolutionary success. Apomictic parthenogenesis, clonal or asexual reproduction without meiosis, is the main or exclusive mode of reproduction, and the telescopic embryo is a peculiarity of viviparous reproduction. This kind of strategy ensures a high population increase during the growing season, reflecting in numerous generations of winged and wingless females. This phenomenon significantly reduced postnatal development time and allows very rapid population growth of aphids. Aphids are highly polymorphic insects with various modes playing different roles in their life cycle. However, oviparous recombined cold-resistant eggs, allowing aphid population to resist harsh winter and to produce an array of new genotypic combinations each year. In this biological context, the sexuales of aphids, oviparous females and males, are the crucial generation in the life cycle of those insects. Animals display a wide variety of sexual dimorphism between taxa, including size, ornamentation, and coloration. In the case of aphids, the most characteristic is sexual size dimorphism. Compared with females, males are much smaller and are slender, with numerous rinaria on the antennal segments, shorter cauda, visible sclerotization on the abdomen, and sclerotized genital structures. Males, winged or wingless, are less numerous and smaller than females. If they are less than 50% of the female size, they are considered to be dwarf. In most species of aphids, polyandry, where one female mates with several males in breeding season, is the main way of the mating system. Aphid life cycles are usually complicated, sometimes with the host plant changes. The typical annual life cycle of aphids starts in the spring when the eggs on the plant hatch, leading to the first generation of aphids. All the aphids born from the winter egg are females called stem mothers. Several more generations of female aphids are born during the spring and summer. A female can live for 25 days, during which time she can produce up to 80 new aphids. Spring and summer reproduction occurs asexual, and newborn aphids are basically clones of the mother. External factors, mainly photoperiod, influence the presence of sexual generation. Thus, in the fall, a generation that grows into both male and oviparous females appear. Oviparous female fertilized by the males lay winter eggs on the host plant, closing the cycle. Some species can have both holocycling and unholocycling population. In warm environments such as the tropics or greenhouse, aphids may go on reproducing asexually permanently. After this so long introduction, there is a question why the life cycle of Svalbard aphids is so amazing and unusual compared to their temperate counterparts. So far, two confirmed endemic species of aphids, Cyrtosiphon svalbardicum and Cytobion calvulum, are known from individual location, as we can hear from Steven that is also a third species. Both of them have an extremely reduced two-generation life cycle. As Cyrtosiphon svalbardicum was an object of my study, I'd like to mainly focus on this species. In the life cycle, the first generation of stem mothers hatching from cold-resistant eggs, usually in early June, and the second generation of sexual forms in July and August, mate and lay eggs before winter. Sexual forms are produced when 24-hour photoperiod, so they are genetically determined. In favorable conditions, like abnormally warm years, the third generation of oviparous females can occur, lead to oviparous females and males, which, however, is not the rule. In the life cycle of this high Arctic species, winged forms are rare, but their increase is also related to a warmer than unusual summer in Svalbard. The biology and various aspects of the ecology of Cyrtosiphon svalbardicum have been studied so far, and this species still is rightly the focus of many researchers. However, the examination of its reproductive system remains unknown. Thus, 3 years ago, I have started a research project, aphids, mainly in the vicinity of the research station in Silesia. Although its host plant, Dres octopetala, is quite common in this area, the aphids were collected only from locations on the east slope of Blandalpytten Cape, west of Nølesund. In summer, adults usually were visible on flower buds on its host plants. However, when I conducted my study at the end of August, the aphids were less numerous and hidden inside the green part of dryas, not easy to find. Successfully, I had collected 15 males and about 100 females, including 2 pairs in copula. Generally, the morphology, histological composition, and ultrastructure of the reproductive system of sexuales of Cyrtosiphon svalbardicum is broadly similar to the reproductive system described already in other species of aphids. The unique characters include in both oviparous females and males an enormous layer of the fat body, adhering to the structures of the internal reproductive system. You can see in the photo as a red ring. The greatly enlarged accessory glands of males accumulate a heterogeneous secretion composed of bunches of spicula-like structures of high electron density material, unknown among any temperate males of aphids studied so far. During mating, this material is transported from the accessory glands to the male to its ejaculatory duct, where it is mixed with the ejaculate and then is transferred to the spermatica of the oviparous female. Histochemical analysis showed that this substance associated with spermatozoa is slightly enriched in proteins. However, we are not sure of the role of these spicula-like structures. Maybe it's some form of sperm protection in response to low temperatures, and definitely requires further study. Because of low dispersal, population of Cyrtosiphon svalbardicum are genetically isolated and mate preferentially with related individuals. According to our studies, the proportion of sexes in late August, at the end of the breeding period and the aphid reproductive activity, was strongly female biased with 1 male per 6 female. Sexuales live singly hidden in the base of the leaves of dryas, which certainly does not make it easier to find a mating partner. As you can see, the Svalbard aphids in particular, better researchers Acyrtosiphon svalbardicum live briefly and must breed quickly to survive. Therefore, they are unique in many respects. Those aphids occur in a few locations in Svalbard and is believed to be endemic. This makes them probably one of the rarest insects in the world. At the same time, Acyrtosiphon svalbardicum is a species with the most north location for any aphid. They are obligatory holocyclic with 2 or 3 generations where fundatrix gives birth directly to sexual mores. The life cycle is shown to be genetically determined and sexual forms are produced when 24-hour photoperiod in temperate aphids is controlled by shortened days. Viviparous wingless or winged females, the dominant generation in temperate aphids, appear in a small number. In favorable conditions, viviparous generation respond rapidly and positively to increased summer temperature, thus this species may benefit from global warming. In those conditions, the additional generation of wingless viviparous females results in the extra sexual generation and a higher number of overwintering eggs, which is the case of Acyrtosiphon svalbardicum, are extremely cold tolerant. In sexual mores, an enormous layer of the fat body adhering to the structures of the internal reproductive system is present. The greatly enlarged accessory glands of male accumulate bunches of spicula-like structures of unknown rule transferred to the spermatica of an oviparous female during mating. Sex ratio are induced by local mate competition and is strongly female biased. The breeding period is about half the length compared with the temperate species. Therefore, the Svalbard aphids really have to feed and breed quickly to survive. At the end, I would also like to mention about one unexpected result of my project, which was the first finding on the aphid species of alien origin in isolated and controlled environment of Svalbard. Mesus persicae is widely distributed and highly polyphagous species I found on 3 different ornamental plants that the owner displayed in front of one of the shops in Longyearbyen Main Street. Plants came from the only market in the archipelago and were imported from mainland Norway. Both the staff at the store and the plant owner were unaware of aphids present, although the colonies of aphids were very clearly visible. Such observation, of course, does not prove that green pitch aphid will be a permanent element of Svalbard's fauna. On the other hand, this species is very flexible and would have a good chance of adaptation in such harsh conditions. Last year, those data were mentioned in the local newspaper Barents Observer about rising threats to the fragile ecosystem of the Arctic. It is a clear signal that our studies are important, which were noticed and shown to the public in the local magazine. The present study was supported by Interact and wouldn't be possible with the help and the hospitality of the whole staff on the research station manager. As we are now a part of SIOS platform, I hope that our study will continue with your kind cooperation. Thank you very much for your listening.
So you should see it by now. This was the first slide. Why geese do or don't migrate to the Arctic. So yes. Basically, I'll tell you why they do migrate. The population we have from barnacle geese on Svalbard is actually coming from Scotland, actually the border between the UK. They spring stage in Norway and they go to Spitsbergen. And the stories from the medieval times were that they came out of barnacles, which is of course not true. They just migrate and they follow the green wave of protein-rich grass. The grasses in general in early spring have the highest protein content. And if you are an animal, you have much more protein in your body as it is than in plants. And so the geese have to be very selective all the herbivores have to be very selective to find protein-rich vegetation. And the digestive system of the barnacle goose is very simple. Here you see the intestine and in the beginning on top the stomach, and actually there are no teeth in the bill, so there's not much fragmentation, and every bite is leaving the body in 1.5 hours. Well, for instance, a reindeer has a complex intestine, a long intestine, a complex stomach, and a blind sac in the end. And food is there for 48 hours. What does this mean? Basically, that in the goose dropping, we can still analyze the diet because the epidermises are still shown under the microscope. The geese don't use cellulose digestion, which is actually the major source of energy for the bigger herbivores. So this is an experiment actually where we do see reindeer eating goose droppings because it's still interesting for them to take the nutrients out of the droppings. And so you see specifically a reindeer chasing the geese away. An experiment I did with René van der Wal is putting droppings in the field and see how fast the reindeer are removing those droppings. We used two types of droppings of geese which were living on a moss diet, which we see a lot laying around, and droppings of geese which are living on a grass diet. We know that because of the diet analysis, but also family birds eat grass and non-family birds seem to eat more moss as that's a safer area. After 2 days, 21 of the grass droppings were removed, while only one of the moss droppings in this experimental setup of 1 square meter, which we repeated 8 times. And this is the average of 8 times. So this clearly shows how important quality of food is for both geese and the reindeer. Of course, the other, so the following the green wave is one of the first reasons. The other reason is that you have constant light from April to August, the whole breeding season, which is very favorable because the geese can then have a rhythm of 1.5 hour feeding, 1/2 hour resting. And I said within 1.5 hour, all the food passes through the stomach. So if they had to sleep for 1.5 hour, they would not digest anymore. Now they are digesting 24 hours a day. Of course, predators are also important in the population process. We did all kinds of integrated population modeling, and it's clear that the foxes are a very important predator. And you can see here data in different years started in 1990. These data are up to 2014, but they do continue. The large variation in predation pressure on goslings, which are born. So we're not talking about the nest, but we're really talking about the grazing period when sometimes all the goslings survive and sometimes none of the goslings survives. So this fits with other studies which state that actually the higher north you come, the less predation there is. This is a Canadian study where they made artificial nests of waders and looked at the predation, and actually the higher latitude gave the longest lifetime of these artificial nests. So the geese have been increasing a lot in numbers. And why did they do so? First of all, protection was very important because there were nature reserves where they could feed undisturbed, but also fertilization of all the areas in the temperate region was important. It meant in the end that geese are not anymore breeding exclusively in the Arctic. Here you see a graph with population increase of actually another flyway of geese, the ones going to Russia. Note the logarithmic scale here, but with this logarithmic scale, actually a constant increase is a straight line. And if the line is increasing, then also the population is increasing exponentially. Or even with the constant line, the population can be increasing exponentially. So we first had geese breeding in the Baltic. And in recent years, there's also a breeding population in the Netherlands, which actually has now the same size as the Svalbard population. So starting only in the 80s, it has now a size which is equal to the Svalbard population, and they breed in the Netherlands. Why? Basically because the farmers did a good job in increasing their production of the grasslands. They put a lot of nitrogen, as you can see in the top left, a lot of nitrogen, which is almost 300 kilograms per hectare. The crops have been more productive. That's in the 2 panels below. And on the panel to the right, you even see that the cattle produce more milk with this increasing quality. And so protein content is not decreasing below a level which is needed for gosling growth. And the other thing is that in the Netherlands, actually, the nitrogen is at the moment falling from the air because of pollution, because of all this nitrogen use in agriculture. It's just the amount of ammonium is now equal to what was the advice for the farmers in the 1960s. So more than 60 kilograms per hectare. We have a big problem in the Netherlands with all this nitrogen falling from the air. But it means that every nature reserve is fertilized too. And so the food has a higher protein content. We looked at the comparison between birds, barnacle geese in Svalbard and birds in the Netherlands. On both slides, you see a lot of trees, but here the trees are a little bit bigger, but the grasses here are also very much grazed. And we took blood samples of these geese and did some tests, some medical tests. First of all, we looked at the white blood cells, what the amount of white blood cells were, but also we looked at the reaction of the plasma in what is called a hemolysis hemagglutination test. If you have a lot of antibodies in the plasma, then actually you can measure that in dilution of rabbit blood and see how the plasma attacks the rabbit blood. That's the analysis for this test. And we did that with the geese in the Netherlands, the temperate region, and the geese in the Arctic. And the red colors are the places where all the samples were located. So on the x-axis, you have a type of white blood cell. On the y-axis, you have the activity in the plasma, and you see that the two populations are really separate, showing more or less that there is more an active immune system in the Netherlands. We did test that with captive goslings. This is on Svalbard, this is in the Netherlands. And the result was that if you look at in the Netherlands, while they looked healthy, but when they were analyzed by a veterinarian, 12 out of 15 had infections, while it was much rarer on Svalbard. Sorry. So to end my story, why geese do migrate to the Arctic, basically it's because of the protein-rich grass in summer, the full day for feeding, and some years with fewer predators. The geese themselves become 20 years, so it doesn't matter if there's a lot of predation in one of the years. And then there's less risk for diseases. But just to link that to why geese, at the moment, some of them don't migrate to the Arctic, then basically that's based on the nitrogen fertilization and deposition, which has changed the quality of the grasses. Of course, there is a night time. There is still a considerable night time of 4 hours, 5 hours in summer. But actually what we do measure is that the geese grow slower. And we also see that in Svalbard, there's actually quite a flexibility in growth speed of the juveniles. It takes then a longer time to go to fledging, but there is more time in the Netherlands as the season is longer. Then there was a lot of predation naturally, but protection and a decrease in hunting has increased the populations and actually makes it also possible for them to breed. And also, of course, the migration itself is very dangerous and might run into problems. So this is why you now can see breeding birds, and even it's now happening in the UK. Last winter, there were 11 birds caught in Calaverock, and 5 of these 11 didn't migrate to Svalbard as it looks right now, but went to Scotland to a local breeding population, which is called the feral population. But we really don't know if these geese are from captive collections or if they really are a change in the populations themselves. So thank you very much for your attention and sorry for going so difficult.
Okay, good. Well, thanks very much, Sridhar, for the invitation, and thanks very much for the very interesting presentation so far. I was sort of asked to give a slightly more overview of ecosystem research on Svalbard. We'll see whether I succeed. what I'd like to cover in the next few minutes is very sort of whistle-stop tour, very brief background on some of the general features of terrestrial ecosystems. We've had some nice introductions to parts of them already, and some comments on so-called biodiversity drivers in these extreme environments. I've got a little bit of a focus of transfers. I'm going to focus on nutrient transfer from sea to land, land to sea, and so on, but also mention other linkages between different types of ecosystems: glacial, terrestrial, freshwater, and marine. And if I get a chance at the end, and I'm glad that Karina has actually raised the question of alien species, I'm going to talk a little bit about human impacts. So I guess you guys are probably more familiar with most of this than me. Svalbard terrestrial ecosystems, what are they? They cover a surprisingly wide range and a surprisingly high diversity. We tend to be familiar with some of the richest of them. So the main picture here is obviously from New Alison. Extensive moss carpets, diverse higher plants. We obviously have the Arctic willow, salix, grazers, grazers, the reindeer have been talked about. Maarten's talked about the geese. We have terrestrial charismatic predators, the foxes, over 150 species of flowering plants. So pretty diverse ecosystems. And Steve has already used this photograph in one of his slides. Just to highlight that the majority of faunal diversity in the high Arctic generally, Svalbard specifically, is of the invertebrate type. You might call it the non-charismatic type. But this is the widest contribution to diversity in terrestrial ecosystems in the high Arctic. You can summarize terrestrial and freshwater food webs in the Arctic. There's a fairly standard sort of diagram illustrating the different elements of the environment: the soil ecosystem, the subsurface ecosystem, the surface system, freshwaters, mountains, screes, glacial ecosystems, and so on and so forth. Lots of components in each of those, all of them playing a role in major fluxes, nutrient cycles, and so on. Polar environments are generally considered extreme. Now, you can sit on Ossian Sars on a nice sunny day and it doesn't feel extreme at all. But year round, you think about the winter climate all of Svalbard experiences. Low temperatures are important, ice formation is important, and generally in more extreme polar environments, physical environmental stresses are considered to be more important drivers or controls on biodiversity and biological processes than our biological drivers. The particular drivers that come up time and time again are water availability, the influence of snow cover, how long can a habitat be active for, temperature, change trends. I should mention nutrients in this bullet, but I did in the next. Across Svalbard, soils are generally poorly developed and nutrient poor. That doesn't mean that there aren't places where they're nutrient rich, but in area terms, the majority of soils are nutrient poor. Large expanses could technically be considered deserts in terms of the overall amount of precipitation they receive on an annual basis. And even if not, typically low air humidities lead to high desiccation stress. Having said all that, various biological factors can clearly be influential, often at smaller physical scales. Nutrient enrichment, I'm going to talk a bit about. Grazing, Maarten's talked a bit about that. Competition, surprisingly poorly quantified, but organisms interacting with each other have various levels of intensity of the interaction. Part of those interactions include predation and parasitism, but they're poorly characterized as well. And then you have human impacts coming in to have a play as well, both the sort of direct form of disturbance, us building on land, digging it up, trampling on it, and so on, fragmenting habitats, and the biological invasions. In terms of the history of Svalbard's biodiversity, if you like, how long has it been there? I'm not going to go through every single point in detail on this. There's a generally accepted view when you compare the Arctic with the Antarctic, the terrestrial biota is relatively rich, but it's also relatively recent origin, gone through multiple glacial cycles. Simplistically, during the glacial maximum, everything is pushed southwards. As the ice retreats, it comes back northwards. So you get these successive cycles of colonization from south to north. That's simplistic, very plainly. There are known major areas of refugia on some of the northern continents. Until fairly recently, though, the assumption was Svalbard had been completely glaciated, everything had been wiped off and had come back again, possibly since the Pleistocene. There is now increasing evidence that's not necessarily the case. Ice-free ground on some of the northern mountains on Svalbard through the last glacial cycle. Where there is ground, there is life. We can demonstrate that today and in far more extreme places than Svalbard. Molecular work on mosses has identified likely refugia on Svalbard and Greenland. And a general feature, if you look at terrestrial ecosystems and the sort of recent paleohistory of those, very rapid post-glacial colonization of these ecosystems and of things like lake ecosystems as the ice retreated, implies that there was a local source for the colonists to come from. We don't simply see, if we try to monitor what's coming in today, we don't see a continual sort of wave of native colonists or additional colonists coming in. So why did they come in very rapidly after ice retreat when they don't come in now? Having said that, as Steve alluded to, I think molecular phylogeography has yet to be applied widely to the Svalbard terrestrial fauna. We don't really know its evolutionary history in as much detail as we often think. Parts of Svalbard, particularly Western Spitsbergen or the western part of the archipelago, are experiencing considerable changes in multiple climatic variables. I think that's well documented. But it does mean that Svalbard is something of a canary in a coal mine for looking at Arctic change responses, both within and at the species level, and at community and ecosystem levels. Important parts of these changes, we always talk about warming, but climate change is far more complex. Changes in precipitation, whether precipitation comes as rain or snow, because that means whether it comes as liquid water or solid ice. Rain on snow event, things that are changing in the winter that have a severe biological impact. And these impacts are observed at various different levels of biological organization, from cellular processes within microbes through to ecosystems and even to biogeography. We need to integrate across all of those. The photos on this slide at the top are of an environmental manipulation experiment run by Kevin Newsham up at New Alesund. And this is looking at how the entire microbial community responds to changes in warming and water availability. From very similar work in the Antarctic, there is a suggestion that this will lead to a step change in the way these communities will function. It's actually testing a very fundamental hypothesis. The two pictures at the bottom are from Steve Coulson, and they're actually of one of the UNIS winter ecology courses a few years ago, and believe it or not, I think, it'll confirm, are of exactly the same site. The idea was to look at how snow cover influenced the underlying terrestrial invertebrate communities. So you can see at the bottom of the bank, you get deeper snow cover, more or less a person deep, than you do at the top of the bank, and that affects the temperature regime. But the point here is to look at the influence of snow cover on underlying soil microclimates. So I will whiz through a few slides on links between, in this case, the sea and the land, nutrient flows I'm really thinking about. This is a very important part of terrestrial biology in the high Arctic. Where do nutrients come from? And a large source of nutrients is the marine environment, and a large proportion of those nutrients come courtesy of marine vertebrates transferring from the sea to the land. They do lead to fertilization, manuring. They also lead to mechanical disturbance, trampling, and the latter and intense polluting levels of manuring actually severely restrict biodiversity within concentrations of marine vertebrates, but this tails off very rapidly with distance from any given colony. In freshwater systems, this leads to eutrophication. So at the smaller scale, you look at point sources of nutrients. I will admit that a whale rib is rather a large point source, but you can see from the top left the influence over time that has had on the development of vegetation community. The skewer at the bottom right is a more obvious point source, and the brightly colored lichens are very characteristic of bird perch locations which get regularly fertilized. So point sources can be small. They can be a lot smaller than this. This could represent a defecation event or a single bird carcass, that sort of thing. But the better known version of this is in the form of vertebrate aggregations. On Svalbard, we're very familiar with highly fertilized bird cliffs and the vegetation growth underneath them. and in the terrestrial environment where bird nutrients are running off the cliff into the surrounding environment. I apologise for the two photos on the left being Antarctic. I simply don't have a nice photograph of a walrus colony, I'm afraid. I'd love to get one day. But elephant seals do much the same thing. Let's go the other way. There has been quite a lot of research on this. It's quite widely appreciated that bird cliffs very plainly put fertilizer into the terrestrial environment, lead to lush development vegetation, lead to denser, more diverse invertebrate communities. And some invertebrates are more or less only found numerically within nutrient-enriched areas. So they're a very important habitat. Having said that, despite the fact it's so obvious, there have been very few attempts as yet to trace exactly how these nutrients get into and flow through these terrestrial ecosystems. There has been a bit more work on this in the Antarctic based on stable isotope work, and this looks at nutrients from penguin colonies being blown around up to a kilometre, basically there's sort of a decay curve up to a kilometre away from colonies. You can trace the nutrients into the plant vegetation, into the grazing invertebrates, into the predatory invertebrates, so you can actually confirm the nutrient flow through the food web. You can also use these vertebrate biodiversity hotspots as predictors of where terrestrial diversity is going to be significantly high. Where else does the nutrient go? I mean, plainly, most of these bird cliffs are close to the sea. Does the nutrients run back into the sea? Is there a sort of completing the circle? That is less clear, but there is Polish research that does suggest that certain elements of the nearshore marine environment are similarly enriched with these nutrients running off the land. And another source of nutrients that is not related to vertebrates at all is the microbial communities that develop in snow. Now, the photograph is from the Antarctic because that's where I had the best photograph of snow algae from. Principal works on glacier surfaces on Svalbard as well. Microbial communities develop. They potentially fix a significant amount of nutrients that potentially runs off into the surrounding terrestrial and marine environments as the snow surface melts. A very poorly quantified contribution to nutrient flows between ecosystems. Melt and freshwater runoff carries things other than nutrients, carries sediments, it carries biological material that can include microbes. So you may be inoculating from one ecosystem to another. This links glacial, terrestrial, freshwater, and marine ecosystems. It's an important source of diversity in glacier forelands. And as I mentioned with the last slide, it's a potentially significant source of fixation and contribution to nutrient and carbon cycles, particularly by glacial communities. Marine vertebrates also transfer some contaminants. They can concentrate heavy metals, both naturally sourced and human sourced, and they can transfer microbial diversity within their feces, basically, which certainly impacts colony soils. Whether it goes any further than that's a different matter. Long-range pollution can be concentrated in these ecosystems. Deposition on the snow surface leads to storage. As the snow melts, leads to outflow. Svalbard has also an industrial history, we all know that, and the concentrated source presented by coal mine spoils leads to acidification, leads to various metal ion leaching, all of which can be traced through the local terrestrial and nearshore marine environments. This is nearly my last slide, so I'm not too far off. We tend to think of Svalbard as a pristine area. A large part of it is under conservation, under intense protection. Having said that, humans are the primary source of new colonists in the polar region. Both north and south, there have been very few records in the last 2 centuries of natural colonists arriving without human assistance and successfully establishing. There have been plenty of records of human-assisted colonists arriving. About a decade ago, there was a survey using brushes and vacuums of people getting off the plains in Longyearbyen, which confirmed multiple viable seeds being imported of plant species that might establish in the natural environment on Svalbard. Having said that, a recent survey by Jess Bartlett and colleagues, published about a month ago, has confirmed that there are relatively few non-native species, up to about 20 species in Pyramidon, I think it is. These are plants, and it is very plain from the data they present that the vast majority, if not all, non-native species are currently limited to settlements, and particularly Barentsburg and Pyramidon. I would have to say there's been very little survey effort on this to date. We don't know the full situation, rather as highlighted by Karina's aphids comment in the shop in Longyearbyen. With the 2 Russian settlements, there is a hypothesized association with the wholesale import of vast amounts of continental soil to both of those settlements, and I'm pretty sure that would come without biosecurity. So, quick overview. Svalbard, well-developed terrestrial diversity and well-developed ecosystems, and it is a key location for understanding multiple aspects of climate change and the impacts on ecosystems. Transfers, nutrient flows, and flows of various other elements are important between marine, terrestrial, glacial, freshwater environments, but they are pretty poorly quantified. This is one of the ways, linking the way these ecosystems function is one of the next most important challenges. There is a need for more integrated approaches across these ecosystem components and drivers. Human impacts can't be ignored. With the number of people and the number of vessels and airplanes visiting Svalbard, there has to be a high priority given to biodiversity and to monitoring and to action in the event of detection of non-native species.